So if you take advantage of using notes for the reading quizzes, what should be on your notes for this particular quiz? You should have at least 3 diagrams on there, right? You have the trip operon diagram, the lac operon, and then the second variation of the lac operon with the cap and the cyclic AMP. Those 3 diagrams should pretty much allow you to answer any questions that can be asked about the operons. Okay? And that would be a good preparation then for material that you will need to know for your exam coming up. So I would encourage you to do that. Anything about lab?
I talked to the folks from Wednesday who need to pick colonies. Those of you who had the lab yesterday, your colonies aren't big enough yet, so you'll pick colonies on Monday and set up your overnights Monday morning, Monday afternoon. Those that had lab yesterday.
Do you have any questions? So we're trying to scratch the surface of talking about the regulation of gene expression in eukaryotic cells. And so far we've talked about a couple of different levels at which we can regulate basically turning on and turning off genes. One of them has to do with chromatin structure. And when we're talking about chromatin structure, basically we're talking about a pretty gross level of regulation, which mainly has to do with what's the state of the DNA and the associated proteins. Is it all folded up, very highly compact, or kind of a loose structure? Why is that relevant? Because ultimately that kind of controls the accessibility of RNA polymerase enzymes to be able to get in and transcribe those genes. The more folded up and compacted that the DNA is, and what do we call that compacted state? In your terminology? Remember, heterochromatin. The more folded up and compacted it is, then the more difficulty there is for those polymerase enzymes to gain access to the genes and to be able to transcribe messages. If the structure loosens up, then we have the euchromatin state. Now it's more easy for those polymerase enzymes to gain access to the promoters of genes and start transcribing. So what determines the state of chromatin? Well, we talked about a couple of main things. One has to do with enzymes that can acetylate histones. Remember, histones are the proteins that cause those nucleosomes to form. And so the more acetylated the histones are, the looser the chromatin structure, and the more transcriptionally active. Methylation of DNA has kind of the opposite effect. When you methylate DNA, it tends to form a more condensed heterochromatin state, less transcriptionally active, and we use the highly methylated X chromosome, the bar body, as an example of DNA that's pretty much permanently inactivated in terms of transcription. So that's one level, chromatin level. Then we talked about at the end of the period last time some initiation of transcription considerations. And they had to do a lot with these control elements and interaction of various proteins with those control elements, which ultimately had a real effect on proteins being recruited or brought to the area of transcription, the promoter particularly, And those particular proteins help to facilitate RNA polymerase binding, which is necessary for transcription to occur. So we talked about the role that activators played. We talked about the DNA bending proteins. We talked about some of these mediator proteins, and then some of these general transcription factors. We refer to these activators as specific transcription factors. Do you want to share the story with them really briefly? You were just telling me about the lactose intolerance.
When you're born, everybody is lactose tolerant, and which makes sense because breast milk has lactose in it. But there's one of these distal control elements that's 13,000 bases, 5' of the coding sequence. So here's the coding, let's say that's the coding sequence for lactase. This distal control element is 13,000 base pairs that way. And in that control element is a cytosine, C. And once you get weaned, that C becomes methylated. And when it's methylated, the transcription factors, the activators can't bind, and you become lactose intolerant because the gene doesn't get transcribed. Those of you who are lactose tolerant, that C is something else. It's usually a T. And the T cannot be methylated, but the transcription factor can still bind. So then you're lactose tolerant. I'm heterozygous. One of mine's C and the other one's something else. I don't know what it is. So the binding of these activators, these transcription factors, to DNA sequences in the distal control element will determine whether or not the structural gene is transcribed. And any mutation in the area of the DNA where the binding factors, where the activation factors are supposed to bind, that affects the binding of those factors will affect the gene.
So the normal scenario is to be lactose intolerant then, right?
Normal is lactose intolerant. But as human populations evolved and became more and more dependent upon milk from cows and goats, it became advantageous for a mutation to occur in the distal control element that would allow for transcription factors to bind throughout your life.
Thank you. Interesting. Did not know that. In addition to regulation of chromatin structure and the initiation process of transcription, there are some other regulatory mechanisms that involve post-transcriptional regulation. Okay, so this is after that particular gene has been transcribed. Remember that gene expression, however, is a product of not just transcription, but then that message that's produced also has to be translated. So there are some post-transcriptional mechanisms for regulation of gene expressions as well. Some of these allow the cell to kind of fine-tune gene expression. So gene expression is happening, but then it can be fine-tuned. And this can give you a very rapid response to environmental changes. If you start regulating expression at the chromatin level or the initiation of transcription level, that's a little bit of slower of a process, and it's not quite as nimble, and it doesn't allow adaptation quite as quickly as these post-transcriptional modifications do. So what are we talking about? Okay, so we talked about chromatin modification, we talked about transcription. One of the post-transcriptional methods of gene regulation is RNA processing. There's some other ones down here in white that we'll subsequently talk about. So we'll kind of go through all of these real briefly. So RNA processing. We've talked about RNA processing already. That was the day I was gone. Dr. Morton talked about this. RNA processing involves this phenomena called alternative RNA splicing. And really what this is involved is when that message is transcribed, if the gene contains introns, those particular introns are transcribed as well. So the message has both introns and exons. Before that message is translated, then those introns have to be removed. And the same message can be spliced in different ways to yield different final messages that are going to be translated. We call that alternative splicing. I think Dr. Morden talked about the troponin gene, didn't you?
That was one of them, yeah.
That was one of the examples you used. So if we've got this troponin gene right here with 5 different exons separated by introns, then this is the primary RNA transcript that's going to be produced from this gene. Everything's copied, introns and exons both. But then when these introns are removed, we can rearrange these exons relative to one another when we form the final product. And so this could be one product of RNA splicing. This could be a different product of RNA splicing. So one set of genetic information can yield several different messages and several different proteins, therefore. When they started the Human Genome Project, which was the attempt to sequence all of the nucleotides in human DNA, at that time, the conventional wisdom was there were about 100,000 genes? Okay. And as they started to do the Human Genome Project, that number kept dropping and dropping and dropping, and I think the current estimate's around 20,000, right? And people were really surprised because 20,000 genes just didn't seem like enough genes to encode all the information you need to make something as complex as a human cell. Well, once they started to figure out that this alternative splicing occurred, then the smaller number of genes made a lot more sense. For example, in Drosophila, there's one gene that has enough exons separated by introns that one gene can generate 19,000 different membrane proteins. Each nerve cell has a different membrane protein. So this gives you a lot of possibilities for variation in the kind of proteins you can produce out of a limited amount of genetic information. It's estimated that about 90% of our human genes, those 20,000 genes, are actually alternatively spliced. So, regulation of mechanisms of alternative splicing can give you ways to regulate gene expression at a different level. How this is regulated, how this alternative splicing is regulated, is well beyond the scope of this course. If you want to get into something like that, Dr. Morden McComb's Advanced Cell Biology course, which he's teaching right now, would be a good bet for you. But this is just another level at which gene expression can be regulated. Once you've got your message, then you need to translate it. Okay? Whether that message is actually translated or not is in part determined by, is that message intact? Okay? Because I told you that all molecules in the cell eventually are broken down. They start breaking up into their amino acids, or their nucleotides in this particular case. We call that turnover. So this represents another potential place where we could regulate gene expression by mechanisms that regulate messenger RNA degradation. How long do these messages last? Do they last a short period of time? Do they last a long period of time? If you can regulate that, you can regulate expression of particular genes. So, lifespan of messenger RNA in the cytoplasm really helps to determine patterns of protein synthesis in the cell. Generally speaking, eukaryotic messenger RNAs have a longer lifetime than prokaryotic messenger RNAs. And one of the reasons is these particular modifications that happen in eukaryotic messenger RNA. And this is something else Dr. Morden talked about when I was gone. The 5' cap and the 3' poly A tail seem to play a very important role in determining how long messages last and what their lifetimes are. In some respects, they're almost kind of like hourglasses stuck on the end of the molecule. And when the sand runs through the hourglass, the message then is signaled for destruction. There's another type of RNA that plays an important role in regulating message lifetime called non-coding RNA. And we're not quite ready to get into that yet. We will in a minute. And actually, I'm going to show you a video about some of these non-coding RNAs. that can actually mark messenger RNAs to be either degraded or inhibited. That particular phenomena of RNAs influencing the degradation of messenger RNA is called RNAi. I stands for RNA interference. We'll get back to that in just a second. So that's another place where we can, regulate gene expression is mechanisms that regulate the lifetime of the messenger RNA. So if the messenger RNA is intact, it can be translated. We can make a protein. So now the gene is expressed, right? We turn genetic information into protein. But some proteins, after they're made on a ribosome, are further processed before they're present in their final active state. Sometimes portions of the polypeptide chain are cleaved off. Sometimes they're chemically modified. A good example of this processing that occurs after translation is insulin. Remember, insulin is a hormone that regulates blood sugar level produced by pancreas cells. So in those pancreas cells, insulin messages are being translated. But before the insulin is in its active form and can be secreted, this proinsulin that's produced by translation has to be cleaved into insulin. And then that insulin that's been cleaved is secreted, moves through the circulatory system, and starts regulating your blood sugar level. So are there mechanisms in the cell that can regulate the cleavage of proinsulin into insulin? Yeah, there are. And then finally, just like the lifetime of a message is important, how long a protein stays around in the cell is important. Okay? If proteins lasted forever, we couldn't really turn them off. The fact that proteins constantly turn over gives us another means of regulating protein levels within the cell. If once they were produced, they just stayed there forever, protein levels would always be high. But then if they start to degrade and break down, if we want to keep them high, we've got to keep remaking them again. Well, there are mechanisms that regulate how long proteins survive within the cell. And one interesting way is that when a cell decides that it's time for a protein to be broken down, it can attach another molecule to it which triggers that protein for destruction. That particular molecule that's attached to the proteins that mark them for destruction are called ubiquitins. And so enzymes that determine whether a protein is ubiquitated, I guess you could call it, would be another regulatory mechanism that would affect gene expression. So, bottom line is that there's a lot of different levels at which gene expression can be regulated. Going all the way from the shape of the chromatin, all the way down to mechanisms that determine whether proteins are properly processed and when they are degraded. This is just scratching the surface, okay? This is a very kind of simplistic understanding of the complexity of regulation of gene expression in eukaryotes. But one of the things I do want you to appreciate is how complicated this is and how amazing it is when you think about it, that because all these mechanisms are working properly at the proper time, in the proper order, we can have something as remarkable as a sperm and an egg coming together to form a zygote and bringing a set of chromosomes from a mom and a dad together to form that potential new child. But we're all starting out as a single cell, 23 chromosomes from mom and 23 from dad. During the process of development of an individual from that single cell zygote, you got to have a lot of cell division, but not all the cells are the same, even though they have the same chromosomes. So as those cells start dividing, they start differentiating and turning into different cell types. And if that particular mechanism doesn't happen in the proper fashion, then we can get horrible defects occurring in the developmental process. If the defects are really too bad, then the developing embryo isn't going to last and it's not going to go on into the fetal stage. If the defects are minor, then we might have a child born, but with certain birth defects. But the fact that we are, once people sitting here are born relatively normal, is really a remarkable thing, given all of the events that have to occur in the proper order at the proper time. And so the development of life really is a miracle. And the more we understand about the molecular basis of all the things that regulate this, the more your mind just kind of goes, it's really hard to wrap your head around that. One of the interesting developments that we have just started to scratch the surface of, probably in the last decade, is the role that non-coding RNAs play in regulating gene expression. Much of your DNA codes for stuff other than protein. It's estimated that of your entire DNA in your cells, only about 1.5% of those A, G's, T's, and C's actually make up genes that code for protein. We call that coding DNA. So what's all the rest of that stuff doing? That's all called non-coding DNA. And it's transcribed. It's transcribed into RNAs. Well, what are some of the RNAs that are produced from non-coding DNA? Well, transfer RNA or ribosomal RNA. But again, that only accounts for a small fraction of the non-coding DNA. What's the rest of these RNAs doing? They're not coding for ribosomal RNA or tRNA. They're not coding for protein. What are they doing? We think they're playing a role in the regulation of gene expression. And they're much more important than we ever thought they were. Some people make the analogy that we used to think that non-coding RNAs were these insignificant little backups, and the messenger RNA and tRNA and ribosomal RNA were really the rock stars of the genetic system. But now we're thinking just that we had that backwards, that these non-coding RNAs, the ones that regulate the whole process, are really the rock stars of the genetic information. So there's a lot of excitement about some of these non-coding RNAs and what they do. One of the things that we've discovered is that these non-coding RNAs can regulate gene expression at the level of translation by either degrading messenger RNA or blocking the translation of that message. So we're making the message, but then these non-coding RNAs are determining whether that message is actually translated into a protein. So this is the level of gene expression that these non-coding RNAs work at. Couple of types of non-coding RNAs that have been studied fairly extensively. MicroRNAs and small interfering RNAs. I've got a picture of microRNAs, but then I'm going to spend most of the time, in the video we'll see in a second, we'll concentrate more on these small interfering RNAs. So both of these are non-coding RNAs that can regulate gene expression. So what are microRNAs? Well, they're small single-stranded RNA molecules that can bind to complementary messenger RNA sequences. Hope you're all comfortable with that concept of complementary. A pairing with U, G pairing with C. So if I've got a sequence of messenger RNA with a bunch of A, G's, and C's, and U's, that's the message. Then these microRNAs would be short segments that were complementary to those sequences in the message. Okay? So this might be a short miRNA. So what happens when these bind to these? It blocks translation, either by just inhibiting the process or by causing this message to be degraded. If we degrade the message, we don't translate the protein, the gene has not been expressed. So these small molecules can inhibit proteins being produced and block gene expression. So here's what a microRNA looks like. It's actually a single strand of RNA folded up into these little hairpin loop-type structures. You've seen something that looks like this before. Remember, transfer RNAs were single strands of RNA folded up into loop-like structures. And this particular region of this mRNA that's transcribed from DNA is one little section of microRNA. There are some enzymes, including an enzyme called dicer, which can chop out these particular sections of microRNA, degrade one of the two strands because they're folded back on one another. And then that microRNA can associate with some proteins and form a complex that attaches to the messenger RNA. So here's the message. It's been transcribed. This complex of protein and microRNA binds by that complementary base pairing and either inhibits translation, maybe blocks it from occurring, or causes the message to become degraded. If your message is degraded, then obviously you're not going to get translation occurring. There's something similar to the microRNA, another class of RNAs called small interfering RNAs. Small interfering RNAs and microRNAs are quite similar. They're just made from different RNA precursors. Small interfering RNAs play an important role in this phenomena called RNA interference. And that's the video we're going to see. It's about a 15-minute video we'll show you that talks about the discovery of this phenomena of RNA interference, or RNAi. Talks about the fact that this is a method of regulating gene expression and how researchers are going to try to use this to study genes and their function and then potentially treat diseases. Something I want you to keep in the back of your minds. This RNAi, these small RNAs, probably evolved as a genetic control system for defense against virus infection by a cell. So it's kind of interesting that we might medically exploit this defense mechanism against virus that evolved in our cells and use it to treat diseases of other types, for example. What's also relevant about this is in lab right now, you are using another evolutionary product of a defense against virus infection in bacterial cells. That defense mechanism is restriction endonuclease enzymes. Those enzymes evolved in bacterial cells to destroy viral DNA that was trying to get into the bacteria and take it over. And we now use those restriction enzymes to create recombinant DNA and do genetic engineering. The evolution of these small interfering RNAs in eukaryotic cells was just another strategy to try to prevent virus from infecting eukaryotic cells. And we're now using that particular strategy and co-opting it to do medical treatments. And it's got a lot of exciting future. So let's take a look at this video. Assuming I can get it going here. It's a little bit of a hokey video, and there's some hokey kind of animation involved in it. But it does a pretty good job of talking to you about the basics of this RNAi and what its potential is in terms of scientific advancement.
Not only are we about to know much, much more about how all these diseases work, Alzheimer's and asthma and arthritis and cancer and HIV and all the others, there's a chance, it's a real chance, that we can treat many of these diseases much more effectively, all because of this one discovery called RNAi, IPM, which I'll explain later. You don't hear much about it, but RNAi is a really big deal. And the curious thing about it is the discovery of RNAi was an accident. It was a puzzle that appeared in a petunia. It was a purple petunia. But to fully appreciate this tale, let's back up a bit. Every creature, and you know this from high school, is made from a recipe. It comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different from mice, and then from whales, and from flowers, but to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big. So big that to RNA researchers like Greg Hannon, RNA is more important than DNA.
DNA really works for RNA, and proteins really work for RNA.
Would you get an argument, by the way, from somebody else?
Oh, undoubtedly, sure.
So how do you get from DNA to become a real creature? Well, let's take one of those fantastic voyages and we'll show you. We're going to find DNA and we'll make it a typical cell, so we're going to have to fly in and then go off to the nucleus of the cell, which will make a beautiful castle, the headquarters. And there's the DNA, the master code inside the nucleus. DNA, says Greg, never leaves the nucleus.
You ever meet one of those mean librarians,
Yeah.
special reserve section?
The ones that go, pow.
Right, you can take the thing, you can copy it, but you can't take the book because somebody else might need it.
So if DNA is locked in the nucleus, how do we get the information out to build our creature? Well, that's what RNA does. That scribe, copying recipes out of the cookbook and throwing them out the window, out to the cytoplasm seed that makes up most of the cell, all those recipes floating through the air, they are RNA. And to finish up in that seed, you see hundreds of thousands, well, we've made them into little guys with chef hats.
Those would be ribosomes, and in your world, there are chefs who are using the recipes that are written in the RNA.
And whenever a recipe lands on a chef, whatever it is, he cooks it.
Whatever it is, he cooks it.
And each recipe is for a protein. Proteins build cells, bone cells, brain cells, all cells. So all these chefs are basically building you. You are made of proteins. And because of RNA, we can copy, we can distribute, and we can cook up you and me. And RNA's been doing this for more than 3 billion years. But there was something spectacular about RNA that nobody knew till just a few years ago, and they learned about it, as we told you, by accident. In 1986, geneticist Rich Jorgensen was working at a biotech startup company in California. He was asked to create a spectacularly dazzling flower.
Looks good.
To attract investors.
So that we could convince venture capitalists, investors, to give us more of the green stuff, more money.
Still, back in 1986, geneticists didn't know how to work that easily with, say, roses. And so
We began with a simple plant, regular garden variety petunias. Petunias being a plant that were easy to introduce genes to in 1986.
And so they decided to create a very, very, very purple petunia. Rich knew which gene produced purple. He knew how to sneak an extra copy of that gene into the plant's DNA, the master text, to be copied by that monk-like scribe.
It will be transcribed by the monk the same as any other gene. And he'll throw the transcript out the window into the cytoplasm, where the chef will be able to pick it up and use it.
Rich thought that if he added more purple recipes, he'd get a purpler petunia. So he did it, and he waited. And what happened?
We produced, instead, white flowers.
White flowers?
Complete opposite of what we had expected. Completely white flowers. We lost pigmentation completely. Our initial reaction was that something must have been wrong with the gene that we had engineered to introduce to the plant.
A mistake.
A mistake. So we checked everything out, and there were no mistakes that we could find.
So why didn't the petunias turn purple? What happened?
The petunia was a big puzzle. Nobody understood why, when you add an extra gene for purple, you should not get more purple, but less purple. It took a decade of brilliant scientists working on petunias and fruit flies and worms and other organisms to finally work out what was going on.
And what was going on is, quite by accident, Rich had discovered a secret inside living cells. Cells from time immemorial have had a mortal enemy called the virus. So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks. But some of those recipes, you'll notice, look a little different. And what's in these recipes is not good for the cell.
No, it's decidedly not good for the cell because the sole purpose of that virus is to make additional copies of itself, and to the point that the entire cell is filled up with this. And the cell explodes, releasing these viruses to go and then infect whatever other cells they can find.
So the theory is that long ago, cells developed a secret defense system which we will call the cop. What the cop does is when viruses invade and create showers of murderous recipes, the cop looks and thinks, some of these have a very fishy shape. It's a chemical difference which comes down to some of the viral recipes are 2 pages instead of 1, and one side is a mirror image of the other. But the point is, to the cop, there's something not right about this shape. So when they see it in that shape,
They say virus. They say,
uh-oh. And the cop destroys the recipe. And when you say it destroys, is this, should we think like kung fu kind of thing? Is it like sort of deal?
yeah, a little enzymatically, a little thermodynamics.
Enzymatically?
Enzymatic kung fu, yeah.
The cop destroys not only the oddly shaped version, whenever he sees that recipe, oddly shaped, regular shape, that recipe in any form must be destroyed to defeat the virus. And the interesting thing is, until 1998, nobody knew that cells had this defense mechanism.
We had no idea it was there. That's what's so amazing, is this whole mechanism had been sitting there where cells were able to tell that something was very funny when they saw mirror image messages and start not just destroying the messages, but destroying anything that looked like that message. They worked out this whole defense system against viral RNA, and we then accidentally stumbled into using it.
The accident was Rich Jorgensen's purple petunia. The question, remember, was when Rich tried to make his petunia more purple, why did it turn white? Well, the answer, it turns out, was that Rich, by accident, discovered the cop. When Rich invaded the petunia cell and inserted his make more purple instructions, he didn't know it, but his purple instructions happened to have that suspicious viral shape. So when the cop saw the recipe, the cop thought, virus, and destroyed every recipe for purple in the cell.
So there's no possibility anymore of producing the purple pigment because the purple transcripts are gone.
If there are no recipes for purple, the chefs don't cook purple.
And because there's no purple pigment produced, the flowers will be white.
And that's how Rich and his petunias helped discover what we now call RNAi.
RNAi means RNA interference, because the cop is interfering with RNA messages, with the recipes in the cell.
And when scientists realized that every plant and animal cell has RNAi, a way to turn off the recipes, turn off genes, they thought, maybe we can use these cops to work for us.
Okay, Trevor?
Which brings us to Marty Russell, 78 years old. She and her husband used to spend lots of time here at their daughter's nursery.
Thank you, Rosie.
Years ago, she enjoyed doing lots of things.
Her passion was reading. She would read everything.
Golf.
Yeah.
I love to play golf, bridge.
But then Marty began losing her sight.
Couldn't see, and I'd probably get the peppers in with the zucchinis, and there would be big problems then.
So she went to her doctor, who told her
You have macular degeneration.
a degenerative disease caused by too many blood vessels growing in the eye, underneath the retina.
As these blood vessels grow, they leak out fluid and blood in the center of her vision, and it's as if you're looking through a very dirty windshield, essentially.
I went home, I was just devastated.
So Marty volunteered to be a candidate for RNAi therapy, something so new she's kind of a pioneer.
She was probably one of the first to get it for any disease whatsoever, and specifically for macular degeneration. Hey, how are you?
Kaiser, how are you?
Good to see you. So this is the VIP room.
I feel very honored.
The reason Marty has so many blood vessels growing in her eye, clouding her vision, is there's probably a mistake in her DNA, in a gene that produces too many recipes that say, make more blood vessels. So the chefs cook up proteins for more, and she ends up with too many blood vessels. Her doctor wants her to have fewer blood vessels. But how do you get the chefs to make fewer blood vessels?
It's pretty easy. You want to shut down a gene? Put in a copy of the gene with its mirror image. Signals the cell, better shut this thing down.
We inserted the needle after numbing her up.
So the doctors literally injected RNA recipes into Marty's eyes that said, make more blood vessels. But they made those recipes look dangerous, like viral recipes, hoping the cop in Marty's cells would leap to it and destroy lots of recipes for more blood vessels, leaving Marty with fewer blood vessels. They wanted the cop to turn off Marty's disease. Did it work?
Marty's vision has improved. It's a very promising result,
I can play bridge.
which is very
Not
important.
great, but, it's part of my life.
She can see flowers again.
Oh, some of them are just gorgeous.
So apparently they did trick the cop in Marty's cells to reduce vein production, although not completely.
I see the yellow. Inside is just a little cloudy, but I can see it.
There's a lot of questions still that need to be answered. This is not a treatment that is proven.
Can we deliver recipes to the right cells?
Lovely.
Does the treatment last?
That's beautiful.
All these are big questions. Still, in mice, RNAi has been effective with Huntington's disease, Lou Gehrig's disease, hepatitis, breast cancer. So says Greg, if we ever work this out in humans,
Any sort of disease that you can imagine becomes fair game.
all the diseases which would be helped if you shut off a gene.
Cancer, HIV, for example.
Wait, is this because you're just an RNA buff that you're saying? You just listed cancer and HIV. These are famous, big, fat diseases.
Arthritis.
Well, stop listing them and tell me, is this a prejudice that you're telling me, or is this true? I mean, these are all candidates for this kind of therapy?
Certainly they are.
And finally, we have saved the best for last. The true power of RNAi goes even deeper than finding cures to terrible diseases. Because what RNAi does, remember, the cop's job is to turn off information, turn off genes.
The big problem of understanding, say, the human genome is you have 20,000 genes. How in the world are you supposed to know what each one does? Well, one very good way to start would be to turn off gene number 1 and see what went wrong.
So you could go through all the genes that make up a human, or for that matter, make up a petunia, and turn off each gene one at a time. If you trick the cop to turn off gene number 1, no color. So gene number 1 is involved in color production. Try gene number 2, no petals. Gene number 2 involved with petals, and so on.
You could make too many leaves, they could curl up, they could be upside down. Almost anything could happen.
But getting rid of the gene tells you what the gene does when it's working.
That's right.
The RNAi discovery is just amazing. 10 years ago, when we were sitting around talking about what would we really need to understand the human genome, we all said we would need some magic way that you could turn off any gene at will, just based on knowing its sequence. And what's happened is this discovery by scientists about RNAi has given us exactly that. It turns out that nature already had a way to turn off any gene at will.
And now, with RNAi as their key, scientists will have the means to decode every living thing, to identify the genes that allow us to grow, that allow us to move, that give us beauty and color. RNA is a modest little molecule, but what it gives us is the world. So now we...
Kind of interesting. I mean, this was just the hot stuff, 5 years ago. And now there's a new hot thing in studying genes and gene regulation and possible gene therapy. Some of you may have heard of CRISPR-Cas9. Dr. Morton has been showing in the lab, right? The TED Talk from the discoverer of that. And that's the new hot area because CRISPR-Cas9 gives us a method to do much more precise regulation and therapy and things like that. The RNAi is a pretty crude method compared to that CRISPR-Cas9. I'm going to talk a little bit more Monday about CRISPR-Cas9 just in general. There's a short video I want to show you. This is something you should be aware of because there are a number of bioethical implications with this particular new technology. So it's something you should be aware of a little bit. So we'll do that. There's probably some stuff you need to talk about in terms of lab next week in the Monday session. And then your LAC and trip opera on quiz. Okay? So have a good weekend. Oh, you mentioned that, yeah. So you're going to sell out? Just remind me, in the fall when you're in micro, because they ask for mentoring usually sometime in the fall. Okay. So, yeah, that'd be cool.